Next up, we have another UT Austin alum, Alexia Martin, who received both a bachelor's in biology as well as a BSA in chemistry from UT Austin, and is now pursuing graduate research in Dr. Rachel Vanette's lab at the University of California, Davis. Lexi researches the gut microbiomes of bees as well as relationships between flower microbes, wildflowers, and bees. And today, Lexi will be talking about acquisition and environmental filtering of introduced floral microbes in the blue orchard bee, Osmia lignaria. Welcome, Lexi.
Thank you, Becca. So yes, my name is Alexia Martin. I'm currently in my 4th year of my PhD, which I don't know how I got there. And yeah, I'm at UC Davis. And before we get started, I also wanted to preface by saying I am more of an entomologist and a microbiologist by training, so please also bear with me. Can I just get a show of hands of people who have thought much about microbes or are really comfortable with thinking about microbes? Okay, cool. So we have a lot of microbe people in here. I was a little worried, but I think we'll be okay. So many people may not know that flowers actually host dynamic communities of microbes within pollen and nectar. So microbes are actually kind of dirty. So here are some flower presses that we did. We basically collected some native wildflowers and then we pressed them onto a petri dish. And you can see all of the variety of and sizes and color of all the different microbes, bacteria, and fungi that have grown as a result. These are just from the flowers. And people have been getting more interested in the last few decades as these methods are becoming more accessible in this concept of a microbiome, which is a group of microorganisms such as bacteria and fungi that live within a particular environment, such as flowers as we see here, or bee guts as I'll be talking about more today. And these microbes aren't just sitting around doing nothing in flowers, they're actually doing quite a bit. So for example, they can interact with each other, such as by competing with each other. They can facilitate each other's growth. There can be priority effects. These are basically super tiny microcosms. They can also interact with floral resources. So this one study found that this bacteria in nectar can actually cause the germination of pollen grains that fall in, which basically breaks open the pollen grain and releases those nutrients into the environment, which the bacteria can then use. And they can also interact with their pollinators. And so pollinators such as bees are actually able to pick up and vector these microbes to different flowers and spread them around. But bee guts can also be a habitat for microbes, which is what I'm super interested in. And a majority of the 20,000 bee species that have been studied to date are solitary, and they pick up a majority of their microbes from their environment. These can come from sources like water, nesting materials that they use like mud or wood, or flowers, which is what I study. And I'm really interested in this question of what impacts the formation of a stable microbiome, which is basically just a super niche community. So hypothetically, factors of the bee gut environment should be able to shape the presence and abundance of organisms within their microbiome. So if we have some community of microbes that are potentially in flowers that a bee is ingesting, then factors of that bee environment, such as the pH of the gut, the oxygen content that's available, or the nutrient contents, should facilitate what's able to grow. And this is a concept called environmental filtering, which also happens in plant communities, so hopefully you're familiar of this in this situation. Oop, we're going back. Okay, forward. And though I think it's really important to investigate the formation of these communities within a microbiome, doing so can be really challenging in complex systems, such as a human gut, for instance. So we can turn to bees as a model system. So I'm using the blue orchard bee. Yesterday, Michael Jones told us about mobs, and today I'm telling you about bobs. And so this is Osmia lignaria, and I would like to plug that I think the reason why entomologists use a lot of abbreviations is actually because the taxonomists give insects such long names. Like, that takes up so many characters to write out. So these bees are solitary. So unlike the bumblebees that we just saw Dylan talk about, each individual bee is basically like a single mom. So she's taking care of all of her own babies, and they nest in cavities. So in man-made situations, we use basically a paper straw that they'll nest in. And the mom will go around to a bunch of flowers. She takes multiple foraging trips and will collect pollen and nectar. And then she'll shape this into this little ball called a pollen provision. And these can be filled with the microbes, potentially from flowers or from other sources that she's interacting with. She'll then lay an egg on the pollen provision. That egg will hatch into a larva. The larva will consume the pollen provision and then will eventually spin a cocoon and pupate. So their life cycle is very similar to like a butterfly. The following spring, the adult bee will emerge and start this process all over again. And this is what this actually looks like. So this is a picture under a microscope. All of this purple is the pollen provision, and this little baby white oval is a baby bee egg. And we know from previous studies that adult blue orchard bee microbiomes are actually environmentally derived because they're super variable between individuals and also across locations. However, so flowers, bee guts, and provisions basically contain the same nutrients if you think about it. So it's just nectar and pollen that these microbes are having access to. But each of these unique habitats may exert different pressures on those microbial communities. So for instance, flowers and their nectar have really high sugar content and low nitrogen content, and petals are subject to UV radiation. Bee guts have a number of host defenses, so insects have immune systems. Guts also have low oxygen content and limited nutrient availability. And pollen provisions, well, the main component of pollen provisions is pollen, and pollen is a really tough nutrient to access because of its tough exine. And this is not even to mention the microbe-microbe interactions that are happening across all of these different habitats. However, studies that have individually surveyed each of these environments have actually found overlap in microbial communities. And so that led me to kind of two main questions. Are flowers a definite source of microbes for adult bees of Osmia lignaria and also for their pollen provisions? And does environmental filtering occur across our three different habitat types? So to test this, we inoculated flowers in a hoop house system with a known microbial community, and then surveyed those microbial communities within each of our habitats before and after our microbial inoculum. And we had kind of two main hypotheses. So one, if flowers are a source of microbes, then we should see an increase in relative abundance of the microbes we inoculated with following our inoculation. And two, if we don't see environmental filtering, then we should see relatively equal numbers of our inoculated microbes across our three environments. But if we do see environmental filtering, then we should see unequal relative abundances of our inoculated microbes across the environments. So we gave our bees Lacey Phacelia or Phacelia tenacitifolia as a food source. I'm sure you guys know a lot more about this plant than I do, so this is just a brief overview. But these plants are annual, and they're native in California, amongst other states. Previous studies have shown that they provide high-quality nectar and pollen sources for many species of bees, including blue orchard bees. And also something that's fun for me is that the pollen is purple. And so whenever I dissect open nest, we can see right here, this is the Osmia nest. These are mud walls that the mother builds herself to divide different brood cells. We've got all this purple pollen, and we have these little tiny bee larvae that are just about to start feeding. And also when I'm looking at the bees, this is a female bee in her nest, and we can see all this little purple pollen on her face, which is very adorable. So the microbes that we chose for this study have known associations with bees and flowers, so we really tried to pick microbes that were specialized within the system. So we used two bacteria. So one is Acinetobacter pollinis. This microbe is able to metabolize nectar and produce volatile compounds, which basically are like smells, that smell very stinky to bees, and bees do not like them, so they'll avoid flowers that are colonized by this microbe. And this is the cool microbe I was talking about earlier that does the pollen germination as well. We also used Apelactobacillus mitchneri, which is named after a famous bee biologist, Charles Michener. And this microbe is found widely within within the guts of wild native bee species. We also used 4 species of fungi and yeasts. However, bacterial and fungal data is processed a little bit differently, so I don't have time to get into all the specifics today, but I'd be happy to talk about it later if you want to talk to me about it. And I also want to point out that these microbes were also not found in previous studies in these same hoop houses, so increases we're seeing should specifically be due to our inoculation and not for any other reason. So what did we actually do? So we started with 2 hoop houses that we seeded with Phacelia tenacidifolia, and at full bloom, we introduced adult male and female bees and provided them with their nesting resources. We then allowed them a couple weeks to get acclimated and start nesting, at which point we took our initial round of collections. We collected adult female bees, pooled flower samples, and marked any nesting progress that had been made up to that point as pre-spray or pre-inoculation. We then did 2 rounds of inoculation. The first, basically what this looked like was we had spray bottles that were filled with water and microbe, and we went up to each individual flower and literally just sprayed them to spray flowers on. And we did this in 2 rounds in order to increase our replicate size since the flowers were only open for about 3 days at a time. We then returned to collect additional female bees, flower samples, and would mark additional nests that had been made as being made post-spray. We used these samples for DNA extractions and sequencing so we could look at the microbes that were there. And then we determined the relative abundance in flowers, adult bee guts, and pollen provisions, and then compared this across our habitat types. So what did we find? Well, for our first question, are flowers a source of microbes for adult bob, females, and provisions? Here we have a relative abundance plot. So on, spoiled my data a little bit there. On the y-axis, we have relative abundance, and then on the x-axis, we have the pre- and post-spray rounds for flowers and guts, or flowers and provisions. For guts, we were only able to collect females before the first spray and after the last spray because we had a limited number of females and we needed them to keep nesting. So for flowers, we saw that there was an increase of one of our microbes in light green, Apelactobacillus mitchneri. In guts, we saw that there was really no difference. And provisions, we saw that there was an increase in both of our microbes. So this suggests that yes, provisions seem to get microbes from flowers, and though they showed up in a couple of gut samples, it wasn't really as common. So this might suggest that that female bees don't have to have these microbes that they're passing to their provisions established in their gut microbiome. Instead, females might just be vectoring these microbes from flowers directly to their provisions. What about for our second question? Does environmental filtering occur across habitat types? So we did see these unequal relative abundances, with the highest abundances being in provisions, followed by flowers, and then in guts. So yes, this also suggests that environmental filtering occurs. And I also wanted to look at this question a little bit more broadly, so not just with the microbes we were inoculating with, but with all of the microbes that we documented in our samples. And so I looked at the alpha diversity. And so we saw that there were really high alpha diversities comparatively in flowers and pollen provisions, and lower alpha diversity in adult bee guts, which suggests that guts might have some additional barriers to colonization. So yes, even more broadly, we do see these signs of environmental filtering. And even if we just look at, these are ASVs, which stands for assigned sequence variants, which are basically just different varieties of microbes. We see that there is some overlap between our 3 habitat types, but mainly each habitat type is dominated by a high proportion of unique ASVs, which may suggest that these microbes aren't able to really establish and specialize across multiple habitat types, but instead might be specialized for one habitat type and then use the other as like a temporary holding space between getting moved to their preferred habitat type. So overall, this study suggests that flowers serve as an acquisition source of microbes and pollen provisions, though gut should be further examined, and also that filtering may lead to unequal establishment of microbes across habitat types. So you might be thinking, how is this more broadly related to bee health, or how is this related to plant health? So flowers are generally considered the dirty doorknobs of bee world. So just how doorknobs are super high traffic areas, and if you have some nasty germ on your hand and you touch the doorknob, then whoever behind you is gonna pick that up too. That's kind of what flowers are for bees. Flowers are super high traffic, and in this study, we saw that these commensal, potentially beneficial, we don't know, microbes are moving through this route, but previous studies have shown that pathogens can also move through this route. And so one example of this is with Crithidia bombi, which is an intestinal parasite that causes bee decline. And it was shown that these can actually be transferred via flowers and bumblebees. But this also happens with plant pathogens. So fire blight can actually be moved between apple trees by bumblebee species. So I hope if you didn't know much about microbes before, I was able to teach you a little bit something new, or you're able to start thinking about microbes in this context. And in the future, I'm planning on looking more into the function of these microbes. Specifically, I'm looking to see if specialized floral microbes offer any sort of protection from pathogens in bee larvae. And lastly, I'll leave you with my acknowledgements. There's a QR code to my website as well as my email, and I'm happy to answer any questions. That's okay. I threw a lot of microbe stuff at you guys.
Amazing talk, thank you. I'm really curious, since we have a lot of restoration practitioners in the audience today, if you have thoughts on how plant conservation efforts should further account for or include consideration of floral microbes within some of these restoration or conservation efforts, if you have thoughts on that.
Yeah, that's a great question. So in general, the kind of dogma of microbial ecology is everything is everywhere but the environment selects. So the microbes are probably just everywhere. I would say, to be honest with you, I do not know much about restoration ecology, so if you guys don't use herbicides or something, which I don't think that you would, that sounds fine, but I think herbicides or fungicides, things like that are applied to flowers, we do know affect these microbes. Yes. Okay.
Yeah, got a question. This is a little bit of a tangent, but it is microbes. The fungi that you found on some of the plants, are any of them documented as kind of being primed for decomposition? In other words, when the decomposition starts to happen of the plant, it's happening quicker than would be normal because it's already been inoculated by the fungi?
Yeah, that's a good question. So in these studies, we can only really identify the microbes to the genus level. It's just kind of where the technology is at right now. So I'm not able to make comments about specific individual species of fungi that are there. As far as the genera that we found, we did find a lot of plant associated, what we would consider plant associated fungi. Decomposition. Yeah, I'm not sure. I could look into the specifics for the microbes. We can talk about that if you would like. Yeah.
I have one final follow-up. Has there been any study into the gut biome of bees that pollinate agricultural fields and maybe the diversity with that versus wild pollinating bees?
Yeah, that's a great question. So generally these agricultural fields are monocultures, right? And so generally studies have found that bees do better when they have a variety of plants that they're feeding from and not just one singular plant. Yeah. Awesome. Thank you very much.